After a carbohydrate is broken down into glucose molecules, there's still the problem of releasing the energy contained inside the glucose. The first step to doing this is the process of glycolysis. So what is glycolysis? Glycolysis is the breakdown of glucose into pyruvate, which occurs in the cytosol of the cytoplasm. The overall process produces 2 ATP molecules, 4 being made and 2 being used, 2 pyruvate molecules, 2 molecules of NADH, and 2 molecules of water from a single glucose molecule. There are 10 enzymes involved in this process. To break the glucose chain, we first have to make it more unstable and easier to break. The enzyme hexokinase helps do this by adding a phosphate group to the chain to produce G6P. Next, the enzyme phosphoglucomutase rearranges G6P into F6P. Phosphofructokinase then transfers a phosphate group to F6P to form FBP. It also regulates the rate of glycolysis, slowing it down or speeding it up as per the requirements of the cell. Aldolase then splits FBP into 2 molecules, DHAP and GAP. While they're both the same molecules in different atomic arrangements, only GAP can move on to the next phase. So the enzyme triose phosphate isomerase converts the DHAP into GAP. Now that there are 2 GAP molecules, every step from now on occurs twice, once with each molecule. The enzyme GAP dehydrogenase, or GAPDH, has 2 jobs. The first is to transfer one of GAP's hydrogen molecules to NAD+ to form NADH. The NADH is dealt with after glycolysis to produce more energy, so we can forget about that for now. The energy from the previous reaction allows GAPDH to transfer a phosphate from the cytosol to the GAP to form BPG. The next step involves adenosine diphosphate, or ADP, which needs to be converted to adenosine triphosphate, or ATP, which is the main way our bodies store energy. To do this, we simply add a phosphate to ADP. The enzyme phosphoglycerokinase does this by transferring a phosphate from BPG to ADP, thus converting BPG into 3-PGA. Since this process happens twice, remember there are 2 molecules of GAP, 2 3-PGA molecules and 2 ATP molecules are produced. Phosphoglyceromutase rearranges each 3-PGA molecule into a different atomic arrangement to make 2-PGA. The enzyme enolase then removes a molecule of water from this to make PEP, which makes it more unstable and ready to lose another phosphate group. And finally, in the last step, pyruvate kinase transfers a phosphate from PEP to an ADP molecule to make ATP. As this happens twice, we end up with 2 molecules of pyruvate and 2 molecules of ATP.